We are going to be talking about how to use BiomeFx to help your patients with mood and neurological concerns. And when we talk about the gut-brain axis, we're talking about how signals from the microbiome travel to the brain and communicate with the brain, and then in return, how signals from the brain travel back to the gut and communicate with the gut. And we're going to be focusing mostly on what's going on in the microbiome and how that is impacting the brain. So our topics for today, we're going to start off with a quick BiomeFx recap. I hope that most of you are familiar with BiomeFx. I won't be going too deep into talking about what BiomeFx is. I'm going to kind of skim over it. But if you do need more information about that, please be sure to review our previous webinars or connect with your rep. I'm going to start off by discussing a dysbiosis and inflammation pattern and then specific markers that are implicated in the gut-brain axis. And then we've got some bonus content, mostly because there's too much content to go over in 1 hour. And I wanted to make sure I shared that information with you. So I probably won't get to talk through the last few slides, but you will have the slides to refer back to. And those are going to be very good resources for you when you're looking for specific patterns on BiomeFx, such as microbiome patterns for the vagus nerve, Parkinson's disease, autism, ADHD, and hepatic encephalopathy. So BiomeFx. BiomeFx is a functional microbiome analysis, and the purpose of it is to provide valuable insight into the gut microbial ecosystem, what is going on in the gut. The meat and bones of BiomeFx is really the functions, and this is telling us what microbes in the microbiome are doing. Are they producing short-chain fatty acids? Or are they metabolizing hormones, for example? And as you will see today, it's really important to not only know which bacteria are in the gut, but what they're doing there, because this is how we know how they are communicating and interacting with other organs in the body, including the brain. BiomeFx paints a picture of the overall strength, resiliency, and health of the microbiome. It's telling us if there's eubiosis or balance versus dysbiosis or imbalance. And this insight is going to direct treatment plans that restore balance to the microbiome and build strength and resiliency. So the functions of the microbes, these are products that they make, byproducts, metabolites. These communicate with extra intestinal organs. I love that word because it sounds like extraterrestrial, but it's extra intestinal organs through what we call the gut organ axis, including the gut-brain axis, which is where we're focusing on today. And we all love the slide, but let's talk about what a healthy microbiome looks like versus a dysbiotic microbiome. A healthy microbiome is going to have nice, healthy, tight junctions, a strong mucosal barrier, a very balanced level of pathogens to commensals, whereas your dysbiotic microbiome has too many pathogens. It's got too many bacterial toxins like LPS. We have a degraded mucosal layer and an inflamed intestinal barrier, and that allows those antigens, food particles, bacterial toxins to leak through that barrier into systemic circulation and elicit an immune response, meaning that we have inflammation now systemically. So how did we get from this healthy intestinal barrier to the dysbiotic inflamed intestinal barrier? There are a number of dysbiosis risk factors that will lead us to this point. And some of these definitely include medications like antibiotics, also stress or lack of sleep, smoking, excessive alcohol, and diet. Diet's a big one that we'll talk about today. So there are a number of these. And how BiomeFx comes into this is that these risk factors, these dysbiosis risk factors, impact the balance of microbes in the gut and lead to dysbiosis. Those risk factors create an environment in which your healthy commensals can't live, and it kills some of them off. And then we have an imbalance. We have too many inflammatory microbes and not enough of our healthy microbes. And because different types of bacteria perform different functions, the postbiotics and functions being performed in the gut are also altered. You can kind of see it as vitamins. Each vitamin has a different function. So if you don't eat the foods that have those vitamins or take a supplement and those vitamins are missing from your life, then the other vitamins can't really compensate for what we're missing out on. And the bacteria balance in our microbiome is very similar. And really, the only way to know how the microbiome has been impacted by those risk factors and therefore how to restore balance is really through a functional microbiome analysis. If I get asked a question, should everyone be on probiotics or should everyone be on the supplement? The answer is you really need to test by using BiomeFx to know exactly what is going on in the microbiome before you make that decision. So who will benefit? Which of your patients would benefit from BiomeFx? It's going to be your patients who have been exposed to those dysbiosis risk factors that I mentioned, and most likely all of your patients have been exposed to those dysbiosis risk factors. From the test, they're going to learn the state of their microbiome as a result of being exposed to those risk factors, and it's going to point to specific diet, supplement, and lifestyle factors that can rebuild their microbiome and improve their health concerns. And specifically today, we're going to be talking about how we can do that along the gut organ axis, the gut-brain axis. It can be helpful for patients with both GI and non-GI symptoms. And today we're going to be talking about how it can be helpful for patients who have anxiety, depression, other mood-related symptoms and conditions not related to GI concerns. From a high level, what you will learn from the report is who is in the gut, what they are doing there, if there is balance, and how to restore balance, strength, and resiliency to the microbial ecology. So I've got a question to start us off with. Will you let me know what is your confidence level interpreting BiomeFx for patients with gut-brain concerns? So if you have a patient with ADHD, autism, depression, anxiety, Parkinson's disease, Alzheimer's, etc., how confident do you feel like you are if you were to pick up their BiomeFx and know how the markers on there correspond to your patient's symptoms? 0 would be you never tried to interpret a test for a patient with neurological or mood concerns. 1 would be you're a little bit confident, and 5 would be that you're proficiently confident you have it. So let me know in the chat what your experience level is with BiomeFx. A lot of zeros. Awesome. Oh, we've got a 5. Good. Okay, so my goal is by the end of this that you're all going to feel like you're at a 5. My second question is, what BiomeFx markers do you consider the most when interpreting BiomeFx for these patients? So if you were to pick up a BiomeFx for a patient and this patient has a gut-brain concern, which markers specifically would you be looking for? We got a GABA. That's one that we will be covering today. Okay. So this is me when I start thinking about the microbiome and open up PubMed. I go down many rabbit holes. We may be going to a few rabbit holes today, but my goal is really to stay away from as many as possible. So I'm going to start off with a very high-level cheat sheet, and I'm going to give you some slides next that are really the most important for you to know. So if you start to get overwhelmed throughout the presentation or at the end of the presentation, then just go back to the next few slides that say cheat sheet on them, and that's where I want you to focus. So let's start with dysbiosis. BiomeFx is going to give you a pretty clear picture of if your patient has dysbiosis. And if so, we can assume that your patient has gut inflammation. And from there, we can assume that your patient has systemic inflammation and that this systemic inflammation is going to impair other organs and systems along that gut organ axis, including the brain. I want you to start off with the basics. If you start off with the basics, you can make a very significant impact. And it is also important after you're comfortable with the basics, then to learn how to specialize a little bit. But even if you are only comfortable with the basics, start there. The basics would include making dietary changes, using total gut restoration. But if I'm going to let you in on a little secret, most of the markers that are imbalanced in gut-brain conditions correlate with having elevated protein in the colon or not enough fiber in the colon. This could be from diet, but it doesn't necessarily mean that your patient's eating too much protein. I often find that it's not a dietary issue when it comes to too much protein. It's more of a digestive issue. So I would first check with diet. How much protein are they consuming? How much fiber are they consuming? And then I would go to digestion. How are they digesting their protein? So if you wanted to focus on something, I would focus on more dietary fibers and better protein digestion. And then learn how to incorporate those specific markers and protocols that we'll talk about today throughout the presentation. So when it comes to the gut brain, what should we look for on BiomeFx? First of all, we're going to look for dysbiosis in the gut, because that means inflammation in the gut, that means inflammation in the brain. And then some of the specific markers that we'll talk about today include LPS producers, short chain fatty acids, indoles, hydrogen sulfide, histamine, ammonia, and GABA. It's important to always take patient history, lifestyle, symptoms, and dysbiosis risk factors into consideration. That seems very obvious, but I have talked to a lot of providers who want to look through a BiomeFx without knowing the patient history. And it makes a big difference, because if you have a patient who has elevated ammonia, for example, elevated ammonia producers on BiomeFx, and they also have a history of an alcohol abuse, then it's going to make that ammonia more significant. Same thing with histamine. The level of histamine that we find on BiomeFx, the significance of that is going to vary greatly according to the patient's symptoms, and if they have symptoms of mast cell activation, for example. I want to start by talking a little bit about what a pattern is, because we talk a lot about patterns, but what does that even mean? And the way that this term, the way that we're using this came about, is that if you do go to PubMed or go to the literature and you search for the gut microbiome and any symptom or health condition, then there's going to be lots of research that has been done, and it's going to be, you're going to look for stool tests. So they've looked for stool tests in these conditions, and as a result, this literature will show a group of bacteria that's elevated in those conditions, a group of bacteria that's low in those conditions, as well as specific metabolites. It might include things like histamine and short chain fatty acids. And that is what we are talking about when we are referring to a pattern. You don't want to take one specific marker and hyper focus on it and give it too much significance. We really want to look at the big picture and what is the pattern that we're seeing overall. So that is what we are referring to as a pattern, and we're going to start off with our dysbiosis and inflammation pattern. These are some markers of inflammation on BiomeFx. There's quite a few of them, starting with low diversity, especially alpha diversity. That means we don't have enough of those beneficial species or enough species overall in the microbiome, which usually allows your opportunistic microbes, pathogens to take up more space and create more inflammation. Also, a low resistome index. So this means that you have high antibiotic resistant genes, which actually activates inflammatory cytokines. Elevated or low dysbiosis ratios. That's a sign of dysbiosis. That's the number one way to tell if your patient has dysbiosis and resulting inflammation. Too many inflammatory microbes. That it could include pathogens. It could also include bile acid metabolizers or elevated proteobacteria and fusobacteria, so that those bacteria that contain LPS. And we'll be talking a lot today about LPS. You can also look for additional gram-negative families in the family section, such as D. sulfibrio. That'll show up on the last page. It's a part of proteobacteria. Elevated inflammatory functions. These are usually functions of proteolytic fermentation. And while they are very important in small amounts to help us to have a nice, healthy intestinal barrier and prevent inflammation, when they are elevated, they're quite inflammatory. And at the same time, having low commensals, so those good, healthy bacteria that are anti-inflammatory, like Akkermansia muciniphila, that produce short-chain fatty acids. So all of this leads to mucosal degradation, intestinal barrier permeability, and systemic immune response. And this is how we get to this unhealthy, dysbiotic, inflamed microbiome that we see in this schematic here. So what do we do about it? This is from my 5 steps for restoring gut health, and it is the presentation that Angelica referred to at the beginning of this when she was introducing me. So the 5 steps I have, number 1, you start off with assessing. And that is what we're talking about today. You use BiomeFx to assess diet, digestion, remove other dysbiosis risk factors, support commensals, and reduce inflammation and rebuild barriers. If you would like more information on how to do this, then you can ask your rep for the recording or find it by logging into microbiomelabs.com, or you can catch my next live presentation will be in just a few weeks. So a basic protocol for this, as far as our supplements would be HU58, Mega IgG 2000, Mega Pre, Mega Marine, Zymbiome Dual, and Mega Quin D3. And I don't have enough time today to educate on why each one of these would be a good choice, but please make sure that you reach out to your rep to ask if you have any questions about these or you're unfamiliar with them. You also want to pull in targeted products for specific symptoms. That might be MegaGuard if your patient has bloating, for example, and then follow up with the total gut restoration. And I'm curious to know how many of you use RestoreFlora HU58 and Megaspore and have preferences for when you would use each. I have my own preferences of when I would use RestoreFlora HU58 or Megaspore. And typically when I'm seeing a BiomeFx for the first time, there are specific markers that I'm looking for that I usually start off with HU58 or RestoreFlora and then move into Megaspore after a few months. So what are your tips for when to use RestoreFlora versus HU58 versus Megaspore? And if you have no idea, feel free to say in the chat box that you have no idea, and please again connect with your rep so they can help walk you through that. Okay, now we're getting to the gut-brain axis, and this is this bidirectional communication system that involves three systems: the nervous system, the endocrine system, and the immune system. Changes in one of the organs will impact the others, and the microbes and their bioactive metabolites in the microbiome are going to influence each of these pathways. This is a little bit about how it works. So here we have the gut lumen. This is where the microbiome is. This is where the microbes are. This is where when you eat food and it's digested, whatever is left over ends up here for these bacteria to eat, and they produce metabolites like short-chain fatty acids. Then we have our intestinal barrier that's separating the gut lumen from systemic circulation. And those metabolites, like short chain fatty acid, are going to move through this barrier, and they can move directly into systemic circulation and then travel all over the body, or they can send signals through the vagus nerve, and the vagus nerve directly connects this gut barrier to the brain. Many of the metabolites that we're going to be talking about today actually work in that way by signaling the vagus nerve to communicate with the brain. And then we have the blood-brain barrier. And these gut metabolites, some of them, like LPS, can actually cross this blood-brain barrier and create inflammation and neuroinflammation in the brain. So a lot of times the same impact they have on the gut barrier, they can also have in the brain. Let's talk about lipopolysaccharide, or LPS. This is a word that you hear us throw around a lot. So what is lipopolysaccharide? It's actually a part of bacteria that are gram-negative. It's what causes them to be called gram-negative. It's the outer cell wall. It's made up of oligosaccharides, and it communicates with the environment, and it does elicit an immune response. We find it in gram-negative bacteria, usually that are more pathogenic, not always, but bacteria like E. coli, Yersinia, Porphyromonas, Pseudomonas, those all have pretty inflammatory LPS. After we eat, especially a meal that is higher in fat, then the gram-negative bacteria die and they release this LPS toxin, and it's really not a problem until this LPS is released. When it is released, it disrupts the tight junctions and it leads to intestinal barrier permeability, so it causes leaky gut. And this is called serum endotoxin because it is a toxin. So after you eat, this process occurs for 5 hours. You'll have more of this toxin leaking through the gut barrier into your bloodstream. This creates inflammation, and we see that by increased interleukin-6, 1-alpha, interferon gamma, increased triglycerides, and postprandial insulin, and this condition is called metabolic endotoxemia. Now, this is normal at low levels, and it can be part of a healthy process if we have low levels of LPS producing bacteria and we have a pretty healthy diet and low levels of inflammation. It should be pretty temporary. However, most Americans, or a lot of Americans, this is a chronic condition because of diet, all those other dysbiosis risk factors that lead to inflammation, and a lot of times an overproduction of these gram-negative LPS producing bacteria. So this leads to chronic subclinical low-grade inflammation. And this is neuroinflammatory and neurotoxic. LPS binds to LPS binding protein, which then binds to pattern recognition receptor CD14 on surfaces of macrophages, monocytes, and dendrites. And as a co-receptor for toll-like receptors, the NF-kappa beta inflammatory pathway is activated. And if you aren't up to speed on all the fancy immune system jargon, this just means that LPS turns on the master switch for inflammation, and we have a big problem, especially if this is a chronic situation. LPS can also stimulate toll-like receptor 4 on the vagal afferent fibers that activate the brain. So the vagus nerve connecting the gut to the brain also has these receptors. LPS binds to them, and it flips on inflammation and activates the brain. This LPS-induced inflammation leads to overactivation of microglia, astrocytes, and dendritic cells, negatively impacting mitochondrial function. And chronic systemic inflammation damages the blood-brain barrier, which in turn allows for LPS, other bacterial products, and neutrophils to enter the brain, resulting in neuroinflammation. So if you remember, this process is also happening in the gut. LPS creates leaky gut and inflammation in the gut, and then it can cross the blood-brain barrier, and it actually causes damage to the blood-brain barrier, and it can cause a leaky blood-brain barrier as well. Chronic neuroinflammation contributes to neuronal damage and synaptic loss, leading to symptoms like memory impairment, muscle weakness, and learning deficits. It also pushes tryptophan metabolism down the neurotoxic quinolinic acid pathway rather than the neuroprotective serotonin-melatonin pathway. So when we eat tryptophan, which is in a lot of protein foods that we eat, that it's very common, it is that tryptophan is metabolized to either a neuroprotective or neurotoxic pathway, and LPS has a lot to do with which pathway it's going to be pushed down, mostly because of this IDO enzyme, which is responsible for conversion to tryptophan to kyurenine, which can result in quinolinic acid. So this enzyme is increased by LPS, but you just need to remember that LPS is going to push tryptophan metabolism down the neurotoxic pathway. And if you aren't familiar with quinolinic acid, the neurotoxic pathway, we do find that it's elevated in Alzheimer's disease patients, and it actually co-localizes with tau and cortical sections. It's increased in the cerebrospinal fluid of suicide attempters, in patients with acute depressive episodes, and in autism patients, as well as a model in rats that they used to model MS. LPS also increases aggregation and accumulation of amyloid beta and tau proteins. These are proteins that aggregate in the brains of Alzheimer patients, forming plaques and leading to neurodegeneration. And here are some of the conditions, the neuroinflammatory conditions that LPS have been correlated to, including stroke and ADHD, anxiety. It actually can help predict depression. The more LPS that we find in systemic circulation, the greater the chance of depression and worse depressive episodes. But really, this is all beginning in the gut. This neuronal dysfunction is beginning with dysbiosis and these symptoms that are occurring in the gut. And some of these symptoms show up well before your symptoms and the brain starts showing up. So you could go as far as to view these GI symptoms of warning signs of brain inflammation. And hopefully, if we're able to recognize that with a test like BiomeFx, we could stop the process before we do start to see those symptoms showing up in the brain. This is what we look for on BiomeFx if we're looking for LPS. We don't measure LPS directly, but what we are looking at is gram-negative bacteria that contain LPS. So if you have large amounts of those gram-negative bacteria, then you're going to have a lot of LPS. This is on the dysbiosis ratio section. These are the 4 predominant phyla in a healthy person: Firmicutes, Bacteroidetes, Proteobacteria, and Actinobacteria. If you've got an imbalance in these ratios, again, if it's high or low, that could mean more inflammation. So we're going to break these down by whether they're gram-positive or gram-negative, starting with Firmicutes in blue, and it is mostly gram-positive. And then we have Bacteroidetes in the turquoise color that's gram-negative. Now notice that the negative sign here is orange because the LPS in Bacteroidetes often is not as inflammatory or toxic as the LPS we find in Proteobacteria, and that's because the structure of the LPS is a little bit different. So Proteobacteria is the main phyla we see that is gram-negative, and it has the worst type of LPS. And then you've got Actinobacteria, which is gram-positive. Some of the pathogens or the bacteria you'll find on the report that are in Proteobacteria include Xylella wadsworthensis, Bilophila wadsworthia, E. coli, H. pylori, Pseudomonas, Citrobacter frundi, Salmonella, and then Desulfibrio is also in Proteobacteria. You'll find it on the family page, and we'll talk a lot about it when we get to the hydrogen sulfide section. And also Bacteroides. There is a pathogen called Bacteroides fragilis that we see often on reports. If you flip back to the phylum page, there are other phylum there besides the 4 that we talked about, and these are some that are gram-negative. The inflammatory potential of these are highly variable, except for Fusobacteria. Fusobacteria, we know, is pretty inflammatory. The LPS that it has is pretty nasty. So for the rest of them, the LPS, the amount of toxicity and inflammation it provides is really dependent on several different factors and not fully understood. So they may be gram-negative, but we don't know how toxic the LPS is that they have. And don't forget that if you are looking for LPS producers on BiomeFx and you see a lot of LPS producers, really high proteobacteria, your go-to is going to be Mega IgG 2000. The immunoglobulins in Mega IgG 2000 binds directly to LPS. It also binds to some pathogens that are gram-negative, like E. coli, Klebsiella, and Salmonella. Next, we are going to move into the function section. So I want to talk about what it means when we talk about functions. And when we talk about functions, we have to start with taxonomy because taxonomy is a little bit different. Taxonomy is using various technologies to identify microbes. We can look for a gene, and we know that this gene belongs to Akkermansia muciniphila, so we have Akkermansia here. Or it could mean that we're culturing and using microscopy to identify something like Candida. On the function section, however, what we're really doing is identifying genetic pathways that code for microbial functions like short-chain fatty acids, histamine, indole, and hydrogen sulfide, regardless of which microbes the genes originated from. When we measure functions with whole genome sequencing, it provides a more accurate insight into what is being done in the microbiome. Because if you're using taxonomy, some tests might try to use taxonomy to assume a function. However, you're really relying on the research that we've done so far to know which bacteria perform different functions. And while there's a lot of research that has been done, there's still so many bacteria out there, and we don't exactly know who they are or what they do. However, if we're looking at functions, we're still able to pick up on the fact that they might have genes that code for histamine production, for example. So we're taking that variable out of it, and we can more accurately assess the functions that are being done in the microbiome. What functions are not? They're not metabolomic. So we're not measuring how much short chain fatty acids are actually in the fecal sample. We don't know how many vitamins are actually in the fecal sample or how that is translating to what we have systemically. We're measuring the bacteria genes there who code for those functions and what those bacteria are capable of doing. So keep that in mind as we go through the next few slides. We are going to start with short chain fatty acid production. And the short chain fatty acids that we're looking at on BiomeFx are butyrate, propionate, and acetate. Short chain fatty acids are metabolites produced by gut bacteria from fermentation of fiber, sometimes protein, and they contribute to GI motility. They're important for blood pressure regulation, circadian rhythms, innate and adaptive immunity, including maturation of microglial cells in the brain. So they are metabolites for gut-brain signaling. And we do know that in certain conditions like Parkinson's disease, Alzheimer's, chronic stress, and anorexia, they tend to be lower, while they're also elevated in obesity and children exposed to chronic psychological stress, as well as autism. And they can be low or high in autism. The important thing there is that there is an imbalance. And this is what it looks like. We've got the gut microbiota. They are consuming the fiber from our diet. As a result, they are producing acetate, propionate, and butyrate, which goes to the intestinal barrier, through the intestinal barrier, and it's impacting these different systems going through the portal vein to the liver and going to the brain, where they play a role in neuronal function. They stimulate the microglia and astrocytes, and they help us to have a nice, healthy intestinal barrier. Again, they help us have a nice gut barrier, and they're important for having a healthy brain-blood barrier as well. I'm going to go through each one of these three individually so you know specifically how they impact the brain. And butyrate activates the vagus nerve and hypothalamus. It does cross the blood-brain barrier. It maintains and restores integrity of the blood-brain barrier by upregulating the expression of tight junction proteins. It promotes the secretion of GLP-1, neurotransmitters like serotonin, and peripheral hormones like ghrelin, leptin, and insulin. And it is anti-inflammatory. It inhibits pro-inflammatory cytokines. These are some of the conditions that butyrate has been studied in. I'll leave this here for reference for you to look back at later. And we'll move into acetate. Acetate is an important energy source for the brain. It's converted to acetyl-CoA, which enters the TCA cycle to produce ATP. It is an alternative energy source for neurons, especially in glucose deprivation, and it's the precursor for acetylcholine, which is the key neurotransmitter involved in cognitive function for learning, memory, and attention. It also influences levels of glutamate, glutamine, and GABA in the hypothalamus. Propionate plays a role in the maintenance of neurons, synapses, and the blood-brain barrier. Its increased levels can lead to propionic acidemia, hyperammonia, motor impairments, and dementia. So propionate is the one that when I'm looking at the short chain fatty acids, if it is elevated, then we may want to dig a little bit deeper and ask about neurological symptoms, because sometimes having elevated propionate is connected to some of those neurological conditions. High levels of propionate induces high levels of microglia activation, neurotoxic cytokine production, abnormal hippocampal histology, and abnormal neurobehaviors. And some studies show that's elevated in autism and Alzheimer's patients. So overall, if you want to increase short-chain fatty acids, most of the time we're more concerned about increasing these short-chain fatty acids, but refer back to the functional fibers table that we have in our recommendation guide. And please make sure that you do have the recommendation guide. It's a separate guide than what you will get when you get the report. So if you don't know what I'm talking about when I talk about the recommendation guide, please ask your rep. But this has different categories of functional fibers in it. A lot of times, even when we think we're eating enough fiber, we're sticking to a few of these categories. By increasing the categories that we're getting foods from, we're feeding different beneficial bacteria, and that will result in increased short-chain fatty acid production. Now we are going to move on to ammonia. Ammonia comes from bacteria urease. So we have this bacterial urease in the intestines, and it decomposes urea to produce ammonia. It's absorbed through the intestinal barrier and delivered to the liver by the portal blood. Ammonia is converted to urea and excreted through the kidneys, but when there is liver disease, there's a decreased capacity for the liver to remove ammonia, leading to elevated blood ammonia levels that then can travel to the brain. The brain is very sensitive to ammonia toxicity, and when it's elevated, it will lead to neurocognitive impairments and altered consciousness. This is what we call hepatic encephalopathy. And here is a pattern for you. We have cirrhosis, so you start off with cirrhosis. The plus sign means these bacteria produce ammonia. So you'll see that there are a lot of ammonia producers in these conditions, and in cirrhosis, we have low diversity along with low short-chain fatty acid production. We've got decreased Bacteroidetes, but we've got increased proteobacteria. Usually there's E. coli, and the activity of the E. coli actually will result in increased Proteobacteria phylum overall, and also increased Fusobacteria, which again is very inflammatory, Enterobacteriaceae, Streptococcus and Villanelliaceae. And then when we progress to hepatic encephalopathy, we also have increased Staphylococcus, Porphyromonas, and actually in the research, when there is Porphyromonas, that it correlates with poor cognitive function. In addition to all this, we find elevated endotoxemia and inflammation, so more LPS. And also with hepatic encephalopathy, there's increased lactobacillus. And that might be a little confusing because we generally think of lactobacillus as being healthier. But remember that it's all about balance in these conditions. And also sometimes we find these, what we would consider healthier microbes showing up in these patterns. And we don't know that they're necessarily causing an issue in those conditions. Sometimes it might just be that the environment is primed for them, or they could even be there as a compensation mechanism as well. If there is elevated ammonia producers on BiomeFx, my recommendation would be that you might want to consider avoiding glutamine supplementation because this could result in more ammonia being produced. It's also important to focus on protein digestion. We want protein digestion to occur before it gets to the colon. It should mostly be absorbed in the small intestine, and the digestion is starting to occur in the mouth with chewing. We need to make sure that we have enough stomach acid. We've got good enzymes being produced, and all those processes need to be working correctly so that we don't have too much undigested protein ending up in the colon. And at that point, it's basically a buffet for these bacteria to consume protein and produce too much ammonia and other metabolites. So work on digestion, support liver health and detox. One of my favorites is Mega Guard. It has bitter compounds from the artichoke leaf extract that help to protect the liver from free radical damage. Mega IgG 2000 can be important for binding the LPS because that LPS is going to also go to the liver and cause some damage. So if you've got elevated ammonia going to the liver, you've got elevated LPS going to the liver, it can be a big problem. So use the IgG to reduce the LPS. And then I like to use Bacillus subtilis HU58, which has been studied in hepatic encephalopathy, and it promotes the growth of non-urease producing organisms, and it also utilizes ammonia for its growth, so it reduces the amount of ammonia in the gut. Next is hydrogen sulfide production. Hydrogen sulfide production produces gas that smells like rotten eggs, and this comes from sulfate-reducing bacteria. It uses sulfate derived from foods containing cysteine and methionine, taurine, and carrageenan. It is important. We do need it because it plays a role in processes relating to inflammatory, immune, endocrine, respiratory, vascular, and neuromodulatory actions. And at low concentrations, hydrogen sulfide produced by bacteria is cytoprotective and maintains gut mucus integrity. So we need it for a healthy intestinal barrier. However, at high concentrations, it leads to mitochondrial dysfunction, damages DNA, and denatures the mucus layer of the intestines. It is elevated in Parkinson's disease, and it may cause alpha-syn aggregation in Lewy bodies. It induces a release of cytochrome C protein from the mitochondrial membrane, reduces ferric ion to ferrous iron, promotes alpha-syn aggregation, and in autism, they find that the severity of autism is proportional to D-sulfibrio, which is a family of hydrogen sulfide producers. So the higher the D-sulfibrio and hydrogen sulfide production, the worse the autism tends to be. This is because this D-sulfibrio binds to iron, it forms iron sulfide, and it can result in iron deficiency, which is also common in autism. And you have to remember that a lot of these hydrogen sulfide producers, like D-sulfibrio, are in proteobacteria, and it has very toxigenic inflammatory LPS, which adds to it. And it may also correlate to schizophrenia. So this is a little bit about how it works in Parkinson's disease. We have an overgrowth of hydrogen sulfide producing gut bacteria, which raises hydrogen sulfide concentrations in the gut cells and in the blood. In the gut cells, excessively increased hydrogen sulfide releases cytochrome C from the mitochondria and increases systolic iron levels. And as a result, the amount of the reactive oxygen species increases. And then D-sulfibrio contains something called magnetite. This is a mineral primarily consisting of iron oxide. And the presence of this magnetite nanoparticles further increases these reactive oxygen species. And then the co-occurrence of the alpha-syn, cytochrome C, and reactive oxygen species, especially hydrogen peroxide, leads to the alpha-syn aggregation. And these alpha-syn aggregates may spread in a prion-like manner to the lower brainstem via the vagal nerve. This is what hydrogen sulfide production looks like on BiomeFx. There are three things to look for here. First is D-sulfibrio, which we find again in the family section. This is just a family of hydrogen sulfide producers. Bilophila wadsworthia is in this family, but you'll find Bilophila wadsworthia on the pathogen page, and it shows up pretty frequently. It is associated with a high fat, high taurine diet, so the Western type standard American diet. And they do see in research that omega-3 fatty acids tends to depress this Bilophila wadsworthia, so it can be almost antimicrobial for this Bilophila wadsworthia while increasing beneficial competing species. There are some other bacteria that produce hydrogen sulfide via the cysteine degradation pathway, including Bacteroides, Enterobacter, Clostridium, Fusobacteria, Klebsiella, Oscillobacter, Prevotella, Porphyromonas, and Villanella. So if you're looking at BiomeFx and you see elevated hydrogen sulfide production and trying to figure out where it's come from, look for these bacteria. It causes gas, bloating, constipation, and diarrhea. So if you have a patient that has D-sulfibrio, Bilophila wadsworthia, increased hydrogen sulfide production, then most likely they're going to have some of these symptoms, and we find it quite often. You may also find patients that think that they are sensitive to everything, but they haven't been able to figure out which foods are causing the sensitivities. A lot of times when they do a BiomeFx and their hydrogen sulfide is through the roof, then we figure out it's not necessarily a food sensitivity, it's that they're feeding these hydrogen sulfide producers. So if we reduce the foods that tend to be a little bit higher in cysteine and protein that feed these hydrogen sulfide producers and decrease the amount of fat or help with fat digestion, then it decreases their symptoms. It can also be associated with SIBO. So here we have in the pathogen section, Bilophila wadsworthia. This is what hydrogen sulfide production looks like on the test. And here is a family page. This is the very last page where this D-sulfibrio will show up. These are the three telltale signs of elevated hydrogen sulfide production. And when you have all of these together, then usually the patient is pretty symptomatic. Where do these bacteria come from? This one's a little bit interesting because it does come from overgrowth of normal flora, like a lot of the pathogens that we find. However, a lot of these bacteria come from the oral cavity. A lot of oral pathogens actually create hydrogen sulfide. And You can also find it in well water. If you've had patients or been around well water that smells like rotten eggs, the reason that well water smells like rotten eggs is because of that desulfibrio. It's in the water. And then patients drink it, and if they don't have enough stomach acid to kill off those bacteria, it ends up in their colon, and then it's creating gas that smells like rotten eggs. So they're very strongly connected. And if you have well water, I do suggest checking it on an annual basis. And I've actually had this show up in clinical practice a few times where a patient had a lot of desulfibrio and BiomeFx, a lot of symptoms, and it turns out they had well water that smelled like rotten eggs as well. So what to do about it? It's important to have a balanced diet that's balanced in fat and protein because many hydrogen sulfide producers thrive on the standard American diet. Improve fat and protein digestion. If they have a perfect diet, they're probably not digesting their fat and protein very well. Decrease cysteine-containing foods like cruciferous vegetables, poultry, and dairy, and use Mega Marine to help decrease that Bilophila wadsworthia. Okay, and now we're going to move on to indole. We talked a little bit about tryptophan metabolism already, but there's 3 pathways that tryptophan from food goes through whenever we consume it. So again, tryptophan comes in a lot of foods that have protein in them, and then it can go down the kynurenine pathway or the serotonin pathway, or it can be degraded by microbes. Most of it goes through the kynurenine pathway, 84%, 5 to 10% down the serotonin pathway, and then the gut bacteria get the leftovers. They get about 4 to 6% of it to ferment and make indoles. This is the same 3 pathways, but here we see some of the bacteria that are involved in the production of indoles from tryptophan. Indoles are strong communicators with the gut-brain axis, and they bind to aryl hydrocarbon receptors in the cytosol, leading to downstream processes like expression of cytokines and modulation of astrocyte activity and neuroinflammation. They act locally in the gut, helping to maintain intestinal mucosal homeostasis, barrier, and immune function, and they act systemically and can cross the blood-brain barrier. They're critical signaling molecules in the communication process of the gut-brain axis. Here we see that indoles have beneficial effects on the intestinal barrier. They impact the liver and the kidneys and can be neuroprotective to the brain, decreasing activation of glial cells and DNA damage by acting as an antioxidant and inhibiting beta amyloid fibril formation, which we often find in Alzheimer's. However, as we see here, while there are many benefits of having a balanced level of indole, elevated indoles can be toxic to many systems, including the intestinal barrier, the kidneys, the cardiovascular system, and the brain by damaging neurons and neural stem cells. Some indoles and derivatives have antioxidant, anti-inflammatory, neuroprotective activities, and nerve regeneration effects. Significant impact on the development and progression of the central nervous system disorders. So we do find that while they're beneficial, elevated indoles may impact and lead to progression of many of these conditions, including autism, MS, bipolar disorder, and schizophrenia. On BiomeFx, this is what indole production looks like. It's hard to create a list of all the different bacteria that produce indoles because there's more than 85 of them, including E. coli, Bacteroides, and Clostridium. If you have low indoles on BiomeFx, this could mean that there's a chance of intestinal barrier permeability and inflammation because we do need them to have a healthy intestinal barrier. Assure adequate tryptophan in the diet. Make sure your patient is eating protein. They're getting nuts and seeds and whole grains. Sometimes intermittent fasting promotes the production of indoles, and ZymBiomeDual may increase tryptophan production directly. However, what we typically see that indoles might be a little bit too high. And whenever this happens, it may contribute to kidney toxicity, oxidative stress in the cardiovascular system, intestinal barrier permeability, and damage to the neurons. It's important to balance protein and tryptophan intake, so make sure they're not getting too much protein. And in the 5 steps to gut restoration, we talked about how much protein your patient should be getting. And the general recommendation is 10 to 35%. That is a very wide range. So you might need to work with your patient a little bit on how much specifically they need, but we don't want a lot more protein than 35% might be contributing to having endos that are too high. At the same time, it's really important to increase fibers in the diet. We need 28 to 34 grams of fiber. Most of us only get half of that. And having healthy fiber protects us from having too much protein, or even if we have poor protein digestion, if we have that fiber there, the good bacteria are going to prefer to eat up that fiber and ferment that fiber instead of the protein, and it will act as a protective mechanism. We also want to make sure we're improving protein digestion and improving any condition that leads to inflammation of the small intestine and may interfere with protein digestion, things like SIBO or IBD. You can also use Zenbiome Dual to push tryptophan down the neuroprotective pathway. If we have too much tryptophan, we want to make sure it's being sent down that neuroprotective pathway and not the neurotoxic pathway. Next is histamine, and I know we're getting close to the end. So I've got histamine and GABA, and then I'll show you the bonus content so you can preview it and review those slides on your own. So histamine, it's an excitatory neurotransmitter and neuromodulator. Now, there are several different sources. There's different places that we get histamine. It can be endogenously produced from mast cells, basophils, and histaminergic neurons in the central nervous system. Our gut microbes can produce some, and that's what we're measuring on BiomeFx. All we're measuring on BiomeFx is what the gut is producing, the microbes are producing. We can also get it from food, like aged cheeses, fermented foods, alcohol, and fish, and from the environment, pollen, for example. Normally, 5% of the total histamine enters the body with food or is produced by intestinal microorganisms. Some bacteria possess enzymes to convert histidine found in protein into histamine. And then we have some bacteria who actually degrade histamine. Histamine is important for modulating the immune response in the gut. It's important for motility and a healthy intestinal barrier. So we do need some histamine, but when it's elevated, it activates the gut immune system through the histamine 4 receptor, which draws immune mast cells into the intestines. And these activated mast cells produce even more histamine, triggering inflammation. It's associated with intestinal barrier permeability. So if your patient has elevated histamine production, then they probably have leaky gut. The immune activation in the gut can influence central nervous system function through cytokine signaling and neuroinflammation. There is research to show that histamine is elevated in several psychiatric disorders, including Tourette syndrome, Parkinson's disease, ADHD, and autism. This is what it looks like on BiomeFx. The major producers include a lot of pathogens and proteobacteria. And really, when we look at the significance of histamine production on BiomeFx, again, we need to take into consideration the patient's symptoms. You can have a very low level of histamine production, and you can have patients that have high histamine type symptoms because it's not just coming from the gut. It actually is possible that you do have patients with maybe hardly any histamine at all showing up, but they do have a lot of mast cell activation symptoms, and that's because it could be more of a mast cell issue than just the gut producing too much histamine. So it doesn't mean that BiomeFx is wrong if your patient comes back with no detected histamine or very low levels of histamine and they have mast cell type reactions. That's completely possible because that is not coming from the microbes. It's coming from those other sources. There are a few things on BiomeFx that you can look at that would make those low levels of histamine react worse in your patients. And the first one would be elevated LPS. If you have a lot of LPS producers, that's going to make histamine reactions worse. If you have elevated hydrogen sulfide and amines, that could also make it worse. Now, elevated histamine means that you have intestinal barrier permeability, and the enzyme that we need to degrade histamine, diamine oxidase, is on the intestinal barrier. So if you have leaky gut, you don't have enough of this DAO supplement or DAO enzyme to break down that histamine. So if it's elevated, resolve dysbiosis and intestinal barrier permeability, eat more fibers and a balanced protein, low histamine diet, consider HU58 and Mega IgG 2000. Mega Marine can also be beneficial. And then you might want additional histamine support, like a DAO enzyme, if you suspect that they may be low on that because they have leaky gut and quercetin. Up last is GABA production. This is an inhibitory neurotransmitter. It's produced from foods containing glutamate. It does not cross the blood-brain barrier. However, acetate can cross the blood-brain barrier and be incorporated into GABA. And gut-derived GABA may act locally on the enteric nervous system or the vagus nerve. So even though it doesn't cross the blood-brain barrier, it's still going to communicate with the vagus nerve, who's going to communicate with the brain. So it still does impact the brain. There is reduced brain and serum levels of GABA in Alzheimer's, autism, and anxiety. It improves cognitive function of patients with neurological disorders, eases anxiety, depression, and stress. And they find that depressed patients exhibit GABA dysregulation. They also have low Bacteroides, which is a producer of GABA. And Bacteroides correlates with decreased brain symptoms of depression. So even though GABA from the gut does not cross the blood-brain barrier, it does seem that GABA production in the gut impacts neurological symptoms. If it's low on BiomeFx, then make sure we're eating fibers to increase short-chain fatty acid production and have more of that acetate. Include foods containing glutamate, mostly if ammonia is healthy. If you have a high ammonia level, you don't want to include a lot of glutamate containing foods because you're going to elevate your ammonia at the same time. So you may want to include foods like tomatoes and mushrooms. Limit caffeine and high-fat foods, as these have been shown to decrease GABA levels. And Zymbiome, lemon balm and L-theanine can help increase GABA production as well. Okay, and then here's our bonus content, and we're out of time. So if you want to go down a few more rabbit holes, then we have some information on bile acid producers in the keystone section and how that can correspond to neurological conditions, as well as a pattern for vagus nerve and the microbiome. We always get lots of questions about the vagus nerve. I have a pattern for Parkinson's disease, as well as autism and ADHD. So a question for you. Now, after listening to this presentation, what is your confidence level interpreting BiomeFx for patients with gut-brain concerns? 1 is you're a little confident, 5 is you're proficiently confident. I took the 0 out, so you don't have that option. Oh, we've got lots of 4s and 3s, 5s. Excellent. Okay, and if you'll tell me, what is one thing that you learned from this webinar that you will take with you and implement in your practice? One thing that you learned that really stuck out to you that you're going to take back with you. And don't forget about the coupon code. And we've also got 2 webinars coming up in May. If you're interested in the 5-step strategy that I referenced, then that webinar is May 8th. And Kara will be here to share becoming a microbiome expert with you. That's going to be the first. So this presentation was the 4th in the series of BiomeFx. And we're starting back over with the first presentation in the series, which will be on May 22nd with Kara.
Thank you so much, Star.
Yes.
All right, so we have a little bit, I know we're over time. Do you think we could go to a couple questions in the Q&A box just to get a couple in before we sign off here?
Yeah, so I see one. What can you do to decrease propionate production? That's a really good question and a really easy answer because propionate is mostly produced by Bacteroides. So if you go back to the dysbiosis ratios page, you'll see Bacteroides. That is the main producer of propionate. So usually when propionate is elevated, Bacteroides is also elevated, and Bacteroides prefers the standard American diet. So cleaning up the diet, including more fibers, making sure that we have good protein and fat digestion and we're not overeating protein or fat, that will help to decrease propionate. A few other things to keep into consideration about propionate is that it is a food additive in some foods, softer foods, so foods like puddings. So if you've got older population that are eating more softer foods like pudding, and they may be on the verge of something like Alzheimer's or Parkinson's, then you're going to want to pay attention to that too, that they could be getting propionate from that source. But the main thing you can do is eat more fibers, make sure that we're not eating the standard American diet. If you change your diet, then it should decrease propionate. What other areas of BiomeFx do you look at for mast cell activation when histamine is low? I would be, if you just go back to that basic inflammation and dysbiosis pattern, that's going to give you a lot of information, because if you've got inflammation in the gut, dysbiosis in the gut, you are going to be stimulating the immune system, which could be creating more mast cell activation. But also look at LPS, hydrogen sulfide, those things. The LPS producers, the hydrogen sulfides, the amines, that will make the histamine tap reaction worse. Okay, I think that's all we've got time for today. Be sure to grab the slides and print those off and use them as you're looking through your BiomeFx reports. I hope this was helpful.